Hello class. Today we're going to review over chapter 8, which includes mitosis and meiosis cell division. So we're going to go through some of these questions here. First, be able to identify and label the parts of a chromosome. So you have this in your notes, and we have gone over it a couple times in class with some different things. But basically we have what's called homologous chromosomes. Homologous. And that's going to be the two together. We have the center spot that's called the chromatid. And I did that wrong. That should be the centromere. Got distracted here. This is the centromere. And this is the chromatid. We have 46 chromosomes, so there will be 46 of those that are in the cell. Define homologous chromosomes. Those are the same pair or leg kind of the chromosomes that are held together by that centromere. When is the genetic information known as chromatin, and when is it called chromosomes? So it's chromatin most of the time. So in interphase, if we're looking at stages, when it is that particular name, interphase would be an example. It's called chromosomes when it forms into these homologous pairs, and it winds the DNA up tight and it makes these chromosomes. So it occurs in cell division, and that's when we'd be able to see them as chromosomes. The normal number of chromosomes for humans is 46. Distinguish between sex chromosomes and autosomal chromosomes. So the sex chromosomes, there are two pair, I guess you'd say. So we have XX is female and XY is male. And then the autosomal, there's 44 of those, and they control some of the other things that are in the cell. So haploid is the half number. For humans, that's equal to 23. Not all other organisms, but for humans. And diploid is the full number of chromosomes, which in humans is equal to 46. What is the purpose of a karyotype? To see the chromosomes. And it helps to see or recognize chromosomal abnormalities. As well as the gender of the individual. All right, so why do cells divide? To grow, to repair, and asexual reproduction. So an organism that's growing is going to have more cells that that need to be made. If you break your arm, you need repair, so you need the cells to repair themselves and make more of them. And then asexual reproduction would be for an organism. An example would be like a bacteria that continues the species itself, then needs to divide in order to have more of them. What does mitosis produce? An example that we had in class was 2N to 2N, so it's diploid to diploid. Or you might also say an exact copy. Okay, what stage do cells live out most of their lifespan? And that stage is going to be in interphase. They generally just live in interphase where they do carry out normal cell functions. They don't have to go into mitosis until the cell needs more of that particular cell. The three stages of interphase are G1, where it grows initially after having been born, kind of. S is synthesis, where it makes DNA. And the reason it makes DNA is only if it is going to go to mitosis. So it will live kind of in this G1 or go through G1, and then it might stop. It might never go through synthesis. G2 is a second growth phase to get ready for cell division. List and describe the stages of mitosis, be able to identify the drawings. So I'm not going to draw them all here, but you can go back in your notes and look at those. List and describe the stages. So mitosis begins with prophase. And due to room, I'll just explain what's there. So prophase, the nuclear membrane disappears, the chromosomes are visible, the centrioles move to the poles, and the spindle fibers develop. And then we go to metaphase, and in this one, the chromosomes line up across the center of the cell. Anaphase, the chromatids are pulled to the opposite ends of the cell. Telophase, the nuclear membrane reforms, the spindles disappear, the chromosomes go back to chromatin, and they start to have a cleavage furrow or a cell plate starts to develop if it's a plant. Describe 3 types of asexual reproduction. So in your notes, I believe, let's see, we had budding. What else did we put in your notes? Binary fission. And spores would be an example. This word here, you'll need to kind of be aware of. Binary fission is basically when we're talking about a single-celled organism like a bacteria going through the stages of mitosis just to make a new organism of itself. All right, the purpose of meiosis is to make gametes, which are the sex chromosomes. Egg and sperm for humans. List and describe all the stages of meiosis. So they're the same as mitosis, except they do it twice. So in meiosis 1, we have prophase 1, metaphase 1, and we have interphase here prior to all that because we have to make a copy of the DNA. Prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1, and then we go to meiosis 2, prophase 2, meta 2, anaphase 2, and telophase 2, and then cytokinesis 2. At this point, we get 4 haploid cells. What do you get out of meiosis? Here we go, 4 haploid cells. Explain the difference between spermatogenesis and oogenesis. So this is making sperm, and you get 4 of them. Oogenesis is making eggs, and you get 1 viable egg and 3 polar bodies. Define synapsis, tetrads, and crossing over. So synapsis and tetrads kind of go together. It's the homologous pairs in meiosis 1 that line up together or match up together. Crossing over is when the tetrads are next to each other in metaphase 1. They, a leg of the chromosomes trade places with each other and allows for genetic variety. The 2 ways genetic variability occurs during meiosis, which is crossing over, which we just described, and then independent assortment occurs during, crossing over occurs during metaphase 1 when they're lined up next to each other. And so you have this chromosome that's lined up here next to another chromosome lined up here. And basically just the legs here trade places and allows for genetic variation. Independent assortment is in anaphase 1. And basically, chromosomes will separate or sort themselves independently of each other. They aren't dependent on one another to determine which way they go, like to which cell they go to. All right, comparing mitosis and meiosis, be able to compare and contrast them, include the differences and similarities in their purposes, process, and final outcomes. So a couple things that we can go through here on this. We did in our notes a couple different ways. We put them in your notes packet and a little chart, but we also made a Venn diagram of them in class, so if you could go back and refer to that. So mitosis has one division, two daughter cells, and it's diploid to diploid. The purpose is growth, repair, asexual reproduction. Meiosis is two divisions, four daughter cells. They are each unique. They are not an exact copy. They are diploid to haploid. And the purpose is to make gametes and for genetic variation or unique individuals. Okay, some problems with mitosis. The last little part that we covered in notes was what happens when mitosis is out of control. And basically what happens is the enzymes don't function properly and it can lead to a tumor. This can be a benign or malignant tumor. So the difference between benign is they are generally healthy cells and they stay in one spot. And so they just make a mass. Malignant, these are cancer cells, and they have the ability to move. Metastasis is when cancer cells move. Possible treatments for cancerous tumors: surgery, radiation, and chemotherapy. Okay, so that's the review of Chapter 8. You will have a unit test over Chapters 2 through 8, so you could go back and just listen to the different review sections as help as well. You do have the review sheet from class, so make sure you look back over that as well.